Okay, so let's say that we are ready to run our t-test. We are going to go ahead and say copy. You're then going to go ahead and put it in a new place. So that is where I'm going to put in my t-test. I'm going to go ahead and copy these two columns since those will help me keep myself straight. The first t-test that we are going to perform is against the baseline. And like I said, each t-test is going to help us look at a different question and analyze the results from that. That is part of what you should be talking about in the discussion when you are talking about the results that you received from the experiment itself. So first we are going to compare the baseline to all the other treatments. The only difference between baseline and all the other treatment is how long they were cultured. So baseline was the starting culture, and the remaining cells were cultured at for 2 days, so 48 or 72 hours, depending upon if you were Monday or Wednesday group. And then obviously you had the various treatments. Some had high serum, some had low serum, some had TMSO added or retinoic acid added. So we are going to go ahead and compare the high serum to baseline, and then we are going to do the rest of those as well. So I'm going to say t-test, and it is going to go ahead and come up with the t-test itself. It's going to ask for your arrays. This is why you needed to do group by group analysis, so everyone was there. So I'm going to be comparing my high serum. So that's the row that I will select all the groups data. Array 2 is going to be what you are comparing it to, in my case baseline. It is a two-tailed distribution, and you are going to be looking at either equal variance or unequal variance. You would have to look that up and test it using the statistical packages here or otherwise. Most of these are probably unequal variance, and I will select that for you here, and you are going to go ahead and press enter, and it gives you the number. Now, a data is significantly different. A t-test is significant if the value that you receive is 0.05 or below. Since this value is significantly above that number, this is not significant at all. Now I'm going to go ahead and repeat that process for the remaining of my samples and go ahead and do the t-test for them. So I'm going to go ahead and do it for low serum compared to, again, the high serum, the baseline. Sorry, we should not be taking the average in here. So 49, and so I'm going to go ahead and reselect it. Yes, I know. Don't complain. Let me do it. So you're going to go ahead and select the area that you want to look at. Again, against the baseline, you're going to do two-tailed distribution, unequal variance, and there you have the number for that. Now you will continue this process for all your t-tests. I'm not going to go through the entire process for you. I'm only doing a couple to show you how you can do this. And for all your various t-tests that you need to perform to look at what the effect of retinoic acid specifically was, or TMSO was, on differentiation, this is the type of process that you would have to perform. So I am going to go ahead and show you this, and then again against your baseline. So you are going to select for that 2, 3, and this one is below 0.05, indicating that it is significantly different. So DMSO does have a significant effect on differentiation compared to your baseline or starting culture. And that's exactly what you are trying to find, is what condition, under what culture conditions, what additions are going to be adding your effect on differentiation, are going to be having the effect on differentiation that you were expecting to find. And this is the last one that I will actually show you. 2, 3. And this one is highly significant because this number is way below 0.05. You are going to repeat that for all the various combinations that we had talked about in class and that are also present in the PowerPoint associated with this. So you can run those and see which ones are significant. If you find a number significant, you want to make sure that you highlight that in your text as well as in your caption related to your table or your graph that you show with it. You can also indicate it on top of the graph by putting in a small star or small cross sign or something on the bar that is showing the significant effect, in this case retinoic acid and DMS. So for the various t-tests, you want to make sure that you compare each one and present each one separately so that we can identify what it is. So if 3 different t-tests show significant effect of retinoic acid, then the bar for retinoic acid should have 3 different symbols on top indicating which t-test show the significant effect. You can also simply show this by showing them in bold or highlighting them into the table itself. Okay, so this is one way that you can do statistical analysis for your data for percent cell differentiation. You can also do the same type of analysis by running an ANOVA analysis. When you do ANOVA, you require to have at least 3 samples per group. Since we have 5 different treatments and we have 3 replicates, we can technically run an ANOVA and compare all the different treatments at the same time using that analysis. All Excel are able to run those for you. You have to add the analytic tool pack to your samples. You can do that by going into File, go to Options, and you will get a dialog box like this. You can then go in and tell it, go into Add-ins, go to the Analysis Tool Pack, and press on Go. When you do, it is going to ask you which one you want. You'll say Analysis Tool Pack and hit OK. Once it is added in, you should be able to see it under Data as Data Analysis. And you can then select it to run the appropriate analyses that you want to run. In our case, the ANOVA analysis.